Hello and welcome everyone. In this podcast, we're going to talk about how the genetic code was deciphered. So deciphering the genetic So remember, we had spoken about the genetic code before that looked something like this, and that how that would predict the number, the amino acids that were being coded for. But how do they go about determining that these various codons coded specifically for a particular amino acid? So let's talk a little bit about that. So before we talk about how they deciphered the code, let's talk about some of the assumptions they made and some of the things they knew beforehand. They knew there were 4 nucleotides in RNA: G, C, A, and U. They also knew there were 20 amino acids. And they knew that these 4 nucleotides had to somehow or another combine in a certain way to make these 20 amino acids. How were they going to do that? Well, if the codon was equal to 1 nucleotide, I'm just going to put NT for abbreviation, then with it being 1 nucleotide, each nucleotide would code for 1 amino acid. So that would only result in a code for 4 amino acids, AA for short, for amino acid. Well, that doesn't seem reasonable. So what if the codon was 2 nucleotides? Well, if you do this quick little formula here, 4 raised to the 2 power, what we're saying is that there are 4 nucleotides here, we know that, and if each codon contained 2 nucleotides, then this would tell us how many different combinations that would make. So 4 squared is 16. So they'd get 16 amino acids. Close, almost enough to make 20 amino acids, but not enough. What if it's 3 nucleotides? Again, we'll go 4 nucleotides raised to the 3rd power. 3, how do we arrange these 4 nucleotides in sequences of 3? How many different ways can we do that? And we get 64 possible amino acids. You could do 4 if you wanted to, and so 4 raised to the 4th power is 256 amino acids. Okay, so if you reason here, we know it can't be 1 because that's not enough. No, it can't be 2 because that would code for 20 amino acids, only 16. It could be 64, or 3 nucleotides, and that would make 64. It could be 4, but that would make 256. So they reasoned, this was just a guess at the moment, that it had to be 3. Because they reasoned that this system has to be efficient. So there's no reason to have 256 codons, or possibilities, when you could get by with 64. And it turned out they were right. And this helps account for the stop codons and the degeneracy we see in the code. And I want to talk about the experiment they did, but I want to talk about it in kind of a very abbreviated manner, and say that they first were able to determine the genetic code for this codon, UUU, AAA, GGG, and CCC. And that's because they knew if they took nucleotides, a pool of, say, uracils, so lots of uracils, and they added this enzyme called polynucleotide phosphorylase, they would get a string of U's. And they knew then that every amino acid that came off of this stretch here would have a codon of 3 U's. And so that this would help us determine that the codon for UUU was a specific amino acid. And the way they used this then was they then took a tube, and inside this tube they placed this string of U's, like so. They then placed a radioactive amino acid. I'll put AA for amino acid. I'll put a little asterisk above it to say it was radioactive. And then they also included what we'll just call translation machinery. So all the tRNAs and all that good stuff. And they did this 20 times with a different amino acid used as the radioactive one. And so UUU, when they put only glycine present, so glycine, no radioactivity. And they did this for all of them. And eventually they came across phenylalanine. And when they put phenylalanine, the amino acid phenylalanine in here, and it was radioactive, with this UUUUU sequence, it became radioactive. Meaning that you were putting together all of these radioactive subunits, amino acids together, to make a large protein. And so they did this to figure out UUU, and then they did it for AAA. And the only thing different was they took a bunch of adenines, they added this enzyme to it, allowed it to make the string of adenines, and then they did the exact same experiment using the amino acids here. So they were able to decipher 4 different codons using this system. Okay, that ends this podcast where we talked about how they deciphered the genetic code. In a quick summary, we showed that they reasoned that the codon had to be 3 nucleotides. The next thing they did was, using that specific enzyme, that polynucleotide phosphorylase, they were able to determine the codon, that these codons AAA, GGG, CCC, and UUU all made a unique, or I should say each made a unique protein sequence of just that amino acid. Now the way they discover the rest of the code, I should have mentioned just real quickly, is that they would add varying amounts of A's and varying amounts of G's and varying amounts of C's and varying amounts of U's and combine a bunch of nucleotides together. However, for our purposes, you just need to know these 4 here and how they determine the code for those 4. Alright, if you have any questions, please let me know. See you in class. Bye.